Sok szeretettel üdvözlök mindenkit. Hát egy kicsivel több hallgatóra számítottunk, bevallom őszintén, de az viszont külön öröm, hogy számos oktató társunk, professzorok is jelen vannak, és érdekli őket ez a mai előadásnak a témája, de hát nyilván annak is örülök, hogy biztos, hogy vannak külföldi hallgatók is a nézőtéren, úgyhogy ha megengedik, akkor a felvezetőt nem két nyelven mondom el, hanem angolul, hogy ne húzzuk az időt, és hogy elkezdhessük ezt, vagy átadhassam a szót a mai délelőtt előadójának, hát Duda professzor úrnak. So, unfortunately, the past 2 years we all had to face a life-threatening pandemic. But we are optimistic, and we are expecting, how to say, a normal academic year without any difficulties. But I guess it's very important to know how did this life-threatening pandemic develop? What is the current situation, and what we expect in the near future. So therefore, it's a particular pleasure to hear about this very important topic from a real expert, a real virologist, Professor Duda, who is a very old friend of mine. And I'm very proud of his friendship, because he was my most important scientific mentor about 30 years ago. So let me introduce Professor Duda, and shortly I will tell you his curriculum vitae, his CV. Professor Ernő Duda was born in Hungary in 1943. He earned his medical diploma from Eötvös Loránd University in 1966, specializing in genetics and biology. Then in 1969, he obtained his PhD in biochemistry and genetics at the Semmelweis University. He was a founding member of the Biological Research Center in Szeged, founded in 1971, where he spent more than 40 years. And during this time, he also done research in several other countries, like in the United States, Canada, France, and or even in Japan. The Hungarian Academy of Sciences, as you know, the oldest and the greatest institution of the Hungarian science, where he achieved first scientific degree, the candidate, in 1977, and then Doctor of Sciences in 1994. Became a professor at the University of Szeged in 1995, and first at the former Department of the Microbiology and Immunology, then the Department of Medical Biology. Besides the University of Szeged, he also taught at Eötvös Loránd University, at Budapest University, Technology and Economics, and also the Sapientia Hungarian University of Transylvania. Furthermore, he became one of the scientific advisor of the Hungarian Academy of Sciences, and also in the Solvo Biotechnology. And after his retirement, he kept working continuously and delivered numerous presentations in regards to the coronavirus and wake of the global pandemic. As a virologist, he ensured the regular and continuous flow of the information while focusing on sharing his research in a comprehensible way for the audience. So, consequence of his hard and persistent work, he received several scientific awards, and in this year, in May, he was awarded to Pro Urbe title from Szeged for his contribution to the medical development of the city. So congratulations on behalf of the faculty of the medical faculty. And of course, I'd like to ask Professor Duda to hold this very, very interesting lecture. Úgyhogy, remélem mindenki élvezni fogja.
Thank you. I felt like being on my burial when they deliver a speech like that. Ladies and gentlemen, if it is winter on the northern hemisphere, it is usually the influenza season. And in the year of 2019, in November, December, in China, there were 400 million people with upper respiratory problems. And usually a small fraction of the infected people develop pneumonia. And there was a Chinese doctor, Chang Chi-Suen, who realized that the computer tomograph picture of some of the patients are very, very different than most of the ordinary pneumonia patients. It had a so-called ground glass look at the picture. And he was frightened, and sooner or later they sent the samples of the patients to a biological lab to sequence the nucleic acid in the sample, looking for bacteria and viruses which might cause this disease. And it turned out that within a few days they sequenced the RNA of the virus, and it turned out that the culprit is a so-called coronavirus. So let's see, what do we know about coronaviruses? Most of the coronaviruses, 4 of them, are ordinary common cold viruses, which usually cause very, very light symptoms, some sneezing, some throat, but after a few days they disappear. Unfortunately, most of these viruses do not elicit a strong immune response, so we can get the same virus every year because there is no really protective immune system. But in 2002, a new virus appeared, a new coronavirus appeared, which we didn't meet earlier. And it was a zoonotic infection. It means that the virus jumped from some kind of animals into humans. Civet cats and bats were probably the original hosts of this virus. And unlike the other coronaviruses, it caused severe respiratory problems in the patients. As you can see, that almost 10% of the infected people died. So it was a very severe virus. Fortunately, as it usually happens with zoonotic infections, it did not spread from human to human, at least not very easily. Usually the infected patients do not produce, though the virus is replicating in the patient, but the patients do not produce enough virus to infect other humans. It was a similar case 10 years later in the Arabian Peninsula when we got another new coronavirus, which was called the Middle East Respiratory Syndrome virus. And that was also a zoonotic, and usually the dromedaries infected the humans. As you can see, the death rate was even worse. Almost 30–40% of the infected people died, but fortunately, practically no human-to-human spread was observed. And then came in 19 the new virus, And we still do not know what kind of animal could be the original reservoir of this virus. The interesting thing with this virus is that approximately 60% of the people who contract the disease, they do not show any symptoms, or the symptoms are extremely light. But there are a lot of people who die because of the infection. And it's genetically very similar to the other coronaviruses, but some of the features of the disease are totally different. Since there is an excellent virology institute in Wuhan where the whole pandemic started, a lot of non-scientists, mostly politicians, mostly American politicians, suggested that it was the virus is not a natural virus. It was created in this virology lab by mean genetic engineering. To answer this question, if it is true or it is really the result of a natural evolution, the first thing what appears to me that who is so idiot to start to make a biological weapon to start with a common cold virus, which in 60% of the patients don't cause any disease. We have so many horrible viruses, so if I want to create a biological weapon, obviously I don't start with a coronavirus. If we scientifically want to answer this question, the best thing is to compare the sequence of this virus to the similar viruses. Obviously, the evolution creates mutations in the sequence of the genome, but these changes are totally random. They do not accumulate at certain places. They happen all over the genome. On the other hand, if I make a genetic manipulation, obviously it affects one part of the genome, and all the changes are concentrated in certain places. And the most important, that it is changing the function of one part of the virus. To show you how it happens, let's say this is the sequence of the virus. And a similar virus is over here. And here we see a missense mutation. Here we see a duplication. Here we see a deletion. So it looks that this virus is obviously the result of some kind of natural evolution. On the other hand, if I see the other virus, in which all the mutations are concentrating in certain places and changing the meaning part of the sequence, there is a good reason to believe that this second virus is an artificial, which was done in a laboratory. So let's see what kind of similar viruses we see in Mother Nature. What you can see here, the data are not the data of the isolation, but it tells you that, for instance, these banal viruses, which interestingly were isolated not in China, these are the closest relatives of the new virus. They were isolated in different caves in Laos. They have approximately 20, 30 years of divergence. So the evolution during 20 or 30 years can create from this virus into another virus. So they are not very, very close in evolutionary sense, but if we compare the genetic sequence of these viruses, you can see this is the present virus, and these viruses are up to 100 years evolutionary distance from them, but the sequence of the viruses are almost identical. There are more than 97% similarity between these two viruses. But the most important thing is that the virus has 4 crucial proteins which are absolutely obligatory to have a good replication. And if we look at the tertiary structure of the proteins encoded by all these different viruses, what we see is that these viruses are exactly the same shape. The proteins are exactly the same shape, and they have the same activity, and they are very, very similar to each other. So on the basis of that, we can say for 100% that this is not an artificial virus, it is a natural virus. When we are talking about dangerous viruses, there are two criteria. One is how much it spread in a human population, how contagious these viruses are. And as you can see, there are horrible viruses, measles or chickenpox, which one person can infect 10 or 20 people. Of course, it depends on the situation of the population. But what is even more striking, if you compare what happens 2 weeks or 3 weeks later, is that in case of measles, within 3 weeks, you can end up with 6,000 patients. On the other hand, as you see, a lot of viruses which we think they are dangerous are not that dangerous at all, because Ebola or SARS-2 or influenza, these are not that much contagious. The other criteria is how dangerous they are, how many people are dying or get a very severe disease. And again, you can see that there is a club of highly dangerous viruses which can kill almost all the infected people. And there are viruses which are in the second league, and they are. So in this respect, I think the human population were very lucky that we got a virus which is not awfully contagious, and it's not awfully a man-eating virus. The strange thing which is characteristic of this coronavirus is that there are certain people who infect a lot of people, while others do not infect anyone. And it turned out during the last 2 years that there are certain people who can produce 100,000 times more virus particle than another person. So obviously there are so-called super spreaders who can infect up to 200 people. In Singapore, they were very careful to trace down who infected whom, and they identified these Mrs. EM, who infected a lot of people, but if you can see, there are a few green-headed figures over here who get infected, but they didn't infect anyone. So this is very, very strange. The other thing is that what happens with the infected people. And it turns out that there are two parameters which are important in this sense. One is the age of the patient. And this is the age trees of different countries or different continents. And as you can see, young people, which means until approximately 55, 60 years of age, the virus practically do not kill anyone. But in the elderly population, the death rate is increasing continuously, and over 80, it can reach up to 20%. Obviously, because of that, the impact of the virus is totally different in aged societies like China, the European Union, or the United States. But the effect in Southeast Asia or in Africa is negligible because there are not too many old people. The other thing is the obesity of the patients. You can see that mortally obese patients are up to 25 times more likely to die because of the infection than lean people who are in the normal range. And again, you can see that the obesity is characteristic of the so-called civilized nations, and it doesn't affect Southeast Asia or Africa. Now, let's go to the molecular biology of the virus. And since it is a single-strand RNA virus, which has a positive strand, it means that it can function as a messenger RNA. And in this case, the ribosomes start to synthesize proteins at the 5' end of the viral RNA, and the first open reading frame results in a polyprotein, and it's a large protein, which interestingly has two kinds of proteolytic activity. So two proteinases are encoded and are present within the polyprotein, and these two proteins cleave the polyprotein at several places, altogether generating 10 functional polypeptides. You probably know that ribosomes are reading always triplets. And it's forbidden to miss the reading frame because it could generate a totally useless protein. Interestingly, in this case, every fourth, every fifth ribosome make an illegal step. And disregarding these stop codon, they read through. And this way they are creating an even longer polypeptide, which, when it is cleaved, it generates up to 16 proteins. And among these extra proteins, which are created by this illegal step, is the RNA-dependent RNA polymerase, which is responsible for the production of the viral RNA, and also the helicase, which helps the polymerase. And this non-structural protein 15 is a peculiar polypeptide because it is a proofreading enzyme, an enzyme which can correct the mutations in the viral genome. The rest of the viral genome codes for the so-called structural proteins, like the membrane protein, the nucleocapsid, the spike, the envelope. These are the red sequences. And we see also these green sequences, which are coding so-called accessory factors. And these are polypeptides or proteins which have a function in helping the virus replication and suppressing the activity of the immune system. The virus altogether codes more than 12 proteins which interfere with our immune system. That's why it can cause a serious disease. In normal, so-called normal cases, if we encounter a virus, once the human cells feel the virus, they start to produce a small protein, I mean a family of proteins which are called interferon. Interferon, the name reflects that it interferes with viral infection. So interferon molecules, when they are produced, they signal to the neighboring cells that prepare for a virus attack. So in these cells, there will be an antiviral state. They cannot be infected with the virus because the cell is producing RNases which destroys the RNAs which enter. Interferons can also elicit the death of the virally infected cells, so the virus cannot replicate in these cells. And the most important function of the interferons is that mobilize the whole immune system. It is a signal which tells the whole immune system that we are attacked and let's prepare for that. This happens in the case of most viruses, but not in the case of SARS-2, because this virus encodes a bunch of proteins which can inhibit the production of interferon, and they can also inhibit the response of the cells to the presence of interferon. So it means that in those individuals where the virus can do the trick, the virus can replicate for several days without alarming the immune system. And in this respect, it's very important that young people especially children, produce a lot of interferon if they are infected by viruses. On the other hand, elderly and sick people do not produce too much interferon, so the virus easily can block the interferon response in these people. And what is a very interesting thing in the patients where the interferon production is mostly inhibited, what we see is that the most typical characteristic of the infection in the clinic is that the people have an increased blood coagulation, a tendency for thrombosis. And the other is that there is an extremely strong inflammatory reaction on behalf of the immune system, so strong response which is damaging the tissue. To see what is the culprit behind it. In the blood circulation, we have different kinds of white blood cells. One type is the so-called neutrophil granulocytes. And these neutrophil granulocytes are the GIs, the foot soldiers of the immune system. They are the first one who encounter the bacteria or the viruses. And they have very powerful weapons to kill them. They can engulf, they can phagocytose the bacteria and the viruses and digest it. They can produce very noxious chemicals which can kill the different kind of pathogenic agents. They can produce free radicals and hypochloric acid. And so it's really a very strong soldier. It has a very peculiar form of suicide, killing the bacteria. It reminds me of the Taliban, who has a bomb belt, and a suicide killer who explodes the bomb and killing everyone around it. The neutrophil can do it by rejecting the nucleus of the cell, all the DNA, all the chromatin proteins, and extremely highly charged molecules, highly positive, highly negative charged proteins. And this trap, which is called a neutrophil extracellular trap, it can stick to all the bacteria and viruses in the environment. So it is a very powerful way to kill them. Unfortunately, when this happens, this clot traps not only the bacteria and the viruses, but also those molecules and those platelets which play a role in the coagulation. And as you can see, all kinds of molecules are entrapped. The platelets are activated, which is a bad sign because it might lead to blood coagulation, but also other compounds are entrapped in this trap. And what is more, it also activates a cascade of proteolytic enzymes which can damage the endothelial cells of the capillaries. And what we can see is that if there are a lot of virus, it creates a lot of nets. And it leads to an overall tendency of coagulation, and the forming microthrombi can do all kinds of problems. They can hurt the kidney, the brain, the heart, and the peripheral tissues. Even in the muscles, it can create anoxia, can create a very bad pain. And as you can see, even the serum of patients who have these netosis, even the serum can drastically increase the frequency of coagulation in healthy people or in the blood of healthy people. The immune system is based on a strategy that it knows what kind of molecular patterns are present in my body. And these are regarded as self, and they are protected. And whatever is not self is non-self. And once the immune system can identify something which is non-self, it attacks it in most of the cases. It's a little bit more complicated, but we have only a few minutes for that. Anyway, if we have a pathogenic agent, a bacterium or a virus, which happens just by accident, it happens to have a molecular pattern, we call it an epitope, which looks like an epitope in my cells, in some of my tissues, then it is a mimicry. It's called a molecular mimicry. On this picture, you can see an insect which looks like a leaf. That is called a mimicry. But there is a molecular mimicry which, in this case, viruses or bacteria carry a lot of epitopes which are very similar of the host organism. And unfortunately, this virus accumulated a lot of epitopes which are very similar to different epitopes in the human body. Normally, the immune system can distinguish these. But when the virus is infecting the different tissues and a lot of cells are dying, so-called danger molecules accumulate, which alert the immune system. And after a while, this tolerance against these epitopes is broken. And the immune system attacks the virus, but at the same time, it attacks different tissues of the host organism. So the virus infection and sometimes the bacterial infection leads to autoimmunity because of the similar epitopes on the pathogenic agent and in the human body. And we think that the long COVID phenomenon, that people who already had the disease, maybe a few months ago, a few weeks ago, a few months ago, they still feel bad. They have very bad symptoms because these, the autoimmunity, so there's antigenic epitopes on the tissues are attacked by the T cells and the B cells, and because of that, the immune system is destroying our own tissues. The next thing I want to talk about is that again, in normal cases, let's say when we are vaccinated, the B cells are responsible for the production of antibody molecules. And these B cells are usually undergo a long process of so-called affinity maturation in the germinal centers of the lymph nodes. In these places, they learn how to recognize the antigenic molecule with very high affinity and very high specificity. And these B cells glycosylate the immunoglobulin molecules. This is just the sugar moiety of the immunoglobulin molecule. And as you can see, there are different kinds of sugars. And the most important thing is that the final maturation is a glycosylation step when a fucosyl molecule is added to the immunoglobulin side chain. Unfortunately, in unvaccinated people, if they get infected, in many people, it's not the germinal center B cells which start to produce the immunoglobulins, but there are so-called extracellular, extrafollicular B cells which are not going through this affinity maturation. They start to produce immunoglobulins. These antibodies, what they produce, they are partially glycosylated, and it doesn't contain any fucosyl molecules. And these molecules, which are produced by these extrafollicular B cells, they have lower specificity, which means that they cannot recognize so well the target molecule, So sometimes they bind to other molecules as well. And they have a lower affinity. So even if they find the virus, they do not stick to the virus at very, very big strength. It turns out that these molecules, which are not fully glycosylated, these are pro-inflammatory. They elicit an extreme immune response, more inflammation. They cannot neutralize the virus very effectively, and because of that, since they have a lower specificity, they can attack much more human proteins in the organism. Part of this business comes in the next slide when we are talking about monocytes, another type of white blood cells. These are also very powerful protective cells. They can phagocytose all kinds of bacteria and viruses, especially those ones which are covered by immunoglobulin molecules. It is called the opsonization. When the immunoglobulin molecules recognize the enemy, that tells the monocytes that this is the enemy, eat it and destroy it. And they usually do it. Unfortunately, when these extrafollicular B cells produce the immunoglobulins and they stick to the virus, the macrophages eat it up because it looks like an opsonized virus. But the protein E, the envelope protein of the virus, can activate a protein inside the monocytes, which is part of the so-called inflammasome. The inflammasome, as the name suggests, plays a role in inflammation. And what happens is the activation of these proteins leads to a programmed cell death, which is called a pyroptosis. And pyroptosis is different than an ordinary cell death, an apoptosis. When cells die with apoptosis, they release a lot of anti-inflammatory molecules in the neighborhood. On the other hand, if the virus induces the pyroptosis of monocytes, they release tremendous amount of pro-inflammatory mediators into the environment, and that is absolutely detrimental if it happens with the alveolar macrophages, because it means that they create an extremely inflammatory environment inside the lung, destroying the lung tissue. So it means that the unvaccinated infected patients those immunoglobulins create this problem. But if a person is vaccinated, so they have already glycosylated immunoglobulins and then get infected, then this problem doesn't happen. During the last 2 years, we were searching for different kinds of medicines, how to treat the patients. But since the virus has an extremely complex replication process, it offers a lot of good targets for medicine development. But unfortunately, developing new medicines usually takes more than 10 years. So 2 years is not enough for that. Obviously, in this case, what we try is we try to reutilize previously developed medicines. And everyone thought at the beginning that since we have so-called broad-range antiviral molecules, which are working very well in case of influenza or Ebola or HIV, so one of these should work with the coronavirus as well. It turned out that they do not, because this non-structural protein 15 is repairing the mutations which are generated in all kinds of other viruses, but not in the coronavirus. So it took 2 years before we came up with a relatively good new nucleotide analog, that is the Paxlovid. But in the meantime, it turned out that usually the real problem is not what the virus is doing, not the virus replication, but it is the immune response, the abnormal immune response which was elicited by different kind of tricks of the virus. In the future, we think, and a lot of research is doing on to inhibit these proteases, because if these proteases cannot work, the virus cannot replicate, because the polyproteins will not be chopped up to functional proteins. The good news is that after a few months, we realized that these extreme strong inflammatory process which kills the patients, the so-called cytokine storm or bradykinin storm, it can be blocked by very powerful steroids or monoclonal antibodies. But these compounds block the immune system. So you cannot use it very early in the infection because it helps the virus. If you are a little bit late, then it's too late. So the timing is extremely important when you start to stop the immune response. And of course, if you stop the immune response, if you modify the immune system, it means that bacteria and fungi and also other viruses might make a super infection. And some of the patients who die because of the infection, they do not die because of the coronavirus, they die because of the super infections. Now finally, a few words about vaccines. Since the viral genome was sequenced in January, I think 12th, so it means that very early in the pandemic, we knew the sequence of the virus, and in many laboratories all over the world, they started to produce vaccines. The thing is that in most laboratories where they started to produce the vaccines, they already had vaccines against Ebola virus and different kind of new viruses. And since they had the system how to do it, it was very easy to change for the coronavirus because just the sequence should be synthesized and put into these vaccines. One type which turned out to be very effective is the so-called vector-based vaccines. These vaccines are using a genetically modified adenovirus, a human or chimpanzee adenovirus. And as you can see, one gene, a so-called early gene of the adenovirus, which is essential for the virus replication, is removed from the virus, so it cannot work, it cannot replicate. And this was replaced by the gene of the S protein, the surface protein of the virus. And what we see is that in this case, this artificial virus gets into the cells start to produce the S protein, so the immunity starts to develop. And once the patient starts to produce immunoglobulins and T cells against the virus, the T cells kill all those cells in which the recombinant virus get in. So besides, the virus, as I said, cannot replicate because a part of the genome is missing, and it can be propagated only in genetically modified cell lines in which this early protein is inserted. Now, it turns out that these vaccines were very, very effective, over 90% effectivity, protecting against death and serious disease. But unfortunately, vaccination with these vaccines leads to antibody production against the adenovector as well. So it means that repeated use of them is not possible because the immune system works against them. The other type of vaccines which became quite famous are the so-called mRNA-based vaccines, which had approximately 10 years life before the coronavirus pandemic, because these vaccines were used for the treatment of patients with malignant diseases. And we knew that they are very efficient, they are very safe. So it was quite evident that immediately those companies which had the technology, they started to produce the vaccines. And it turned out that they are exceptionally effective and exceptionally safe, almost no side effects. And they had close to 100%, they provided close to 100% protection against severe disease or death. The structure of these vaccines is a nanoparticle in which different kind of modified lipids entrap the RNA of the S protein, coding for the S protein. And the thing is that once the messenger RNA is introduced in the cell starts to read it and produces the S protein. It elicits the immunity. But the messenger RNA is digested within a few hours. It disappears. The lipids are digested. They disappear. So one day after the shot, there is nothing related to the original injection which remains in the cells. Besides, those cells which are starting to produce the S protein, they express the S protein on the surface. So once the immunity is developed, the immune cells are killing those cells. So it means that 1 week after the shot, absolutely no sign of the injection, no molecule which has anything to do with the original vaccine. And the take-home message is that these excellent new type of vaccines prove to be so safe and so effective that all other type of traditional vaccines became almost totally useless. And right now, only the messenger RNAs are used, and probably in the near future, we will not see too many other type of vaccines. Perhaps there is an American company developing a nasal spray with a so-called leaf vaccine, which means that a replicating virus, which is an attenuated virus, and they hope that it could give a mucosal immunity which lasts a little bit longer, because right now the main problem with the vaccines is that they cannot trigger a long-lasting immunity. It cannot protect us for maybe 8 or 10 years like a hepatitis B virus vaccine. Thank you for your attention.